Okay, here's part 2 of our digestive system lecture. So we have the liver. The second, just the largest gland of the body is the liver. Here's that falciform ligament that we talked about, a serous membrane, F-A-L-C-I-F-O-R-M, which attaches the liver to the diaphragm and the anterior abdominal wall. So the diaphragm is going to be up here at the top, the muscle up here, and then this is the front of the abdominal wall here. So that falciform ligament helps to secure the liver. The functional cells of the liver are called hepatic cells, H-E-P-A-T-I-C cells, and they form bile. Now you can see over here I've boxed in the word hepatocytes. So all these little brown cells here on this picture are hepatocytes, and they will form bile. Now we have these lobules, and here's a lobule up here in the upper left. This is a lobule. You see it's a circular arrangement, has radial cords that come out from the center, from the central vein out. Lobules consist of radial cords of hepatic cells arranged around a central vein. There's a central vein in the middle, and you can see a boxed in central vein here on the picture on the right, but you can also see it on the picture on the left. The central vein is in the center of a lobule. Now capillaries over here on the right hand side. You're going to have capillaries, or little vessels that are coming in from your hepatic portal vein. This is a vein that's picked up blood from your digestive tract that contains nutrients, and a branch off the common hepatic artery. Remember the celiac branches into the common iliac, the left gastric, and the splenic. Well, this is a branch off of the common hepatic, and you see that they both dump blood into this lobule into an area called a sinusoid. And here it is. I've got a box over here on the left. It says capillaries enlarge to form vessels called sinusoids, S-I-N-U-S-O-I-D, which are lined with Kupffer cells. That's an old name. That's a person's name, so I wrote over here on the side. Stellate reticuloendothelial cells. Those are the same thing as Kupffer cells. Now those phagocytic cells are there to make sure that that fluid containing nutrients that was absorbed from your digestive tract doesn't contain any foreign pathogens. And so it's there to help to make sure that that blood is clean as it goes through this sinusoid toward the central vein. Bile is secreted by the hepatic cells, again going back to the hepatocytes here, into bile canaliculi, B-I-L-E. Second word is C-A-N-A-L-I-C-U-L-I. Iculi means little, so it's little canals, little bile canals. So you can see on the picture where you have the green bile and the bile canaliculi. I have them in a box here. Bile in bile canaliculus flows into bile duct. So bile is produced by the hepatocytes, and the bile moves to the outside of the lobule where the little bile duct is. Now it says, which channel bile into small ducts which eventually become the right and left hepatic ducts. So let's look at this picture. Here's the right and left. That should be hepatic ducts, H-E-P-A-T-I-C, ducts. So by looking at this picture and seeing that the one on this side of the picture, the left side of the picture, is actually the right, the gallbladder is on the right side of your body, the right side of the liver. Okay, so the right and the left hepatic ducts are going to come together. It says these bile ducts, these should be these hepatic ducts, join together to form the common hepatic duct. So the right hepatic duct and the left hepatic duct join together to form the common hepatic duct. Now let's look at something before we continue. Over here on the right is your gallbladder, And the gallbladder is a sac which stores excess bile for a future meal that has a lot of fat in it. And it has a duct called the cystic duct, which leads to it. So in your notes, the common hepatic duct and the cystic duct are going to join together. So the common hepatic duct and the cystic duct join together to form the common bile duct. That should be common bile duct, C-O-M-M-O-N, bile duct. The common bile duct, you see it goes on down, and you see here's the pancreas. It has several pancreatic ducts. So the common bile duct and the pancreatic duct joins together to form the hepatopancreatic ampulla, H-E-P-A-T-O-P-A-N-C-R-E-A-T-I-C. Second word is ampulla, A-M-P-U-L-L-A. An ampulla is a swollen structure. So the combined vessel is kind of swollen, which empties into the duodenum, the first part of the small intestine, by way of this little bump at the end of the hepatopancreatic ampulla where it enters into the small intestine, the duodenal papilla. A papilla is a projection, right? And it's in the duodenum in the small intestine, so they named it the duodenal papilla. If you were to look at that, it looks just like a little mosquito bite on the inside lining of the duodenum. Now you have a picture in your notes, and here is the picture. I've got it all labeled for you in order. So let's look at the numbers. Number 1, the right hepatic duct, and there's the left hepatic duct, number 2, joined together to form number 3, the common hepatic duct, and 3 and 4, the cystic duct, joined together to form number 5, the common bile duct. The common bile duct number 5 joins to number 6, a pancreatic duct, to form a swollen vessel called number 7, hepatopancreatic ampulla, which empties into the duodenum by number 8, the duodenal papilla. So I've got your picture labeled for you, and you can just go through that sequence again in the same order. It even points out the falciform ligament up there. Remember, the falciform ligament is attached to the diaphragm and the anterior abdominal wall to help support that liver. Now we have some, back to the liver again, on the blood supply. This is on this slide. The hepatic artery, this is the branch of the common hepatic artery, delivers oxygenated blood from the aorta to the liver. Okay, so it's aorta, celiac, trunk, and then the common hepatic is coming off. This is a branch off the common hepatic, so it does contain oxygenated blood. The hepatic portal vein, H-E-P-A-T-I-C, second word portal, P-O-R-T-A-L, vein carries deoxygenated blood containing nutrients from the digestive tract. So they're both going over here to the liver. The hepatic vein empties blood from the liver into the inferior vena cava. So after all this blood is filtered by the sinusoids and it goes to the central vein there, you see the central vein on the picture on the left, it says interlobar veins to the hepatic vein. All those are going to form the hepatic veins which empty from the liver into the inferior vena cava. So going back again, blood from the hepatic portal vein and the hepatic artery, the little blue and the red vessel here, enters into the sinusoids of the liver, the swollen vessels called sinusoids. The blood carries oxygenated nutrients to the hepatic cells. That's how they get their blood supply and their oxygen. Remember, the portal arteriole is coming off of the aorta. And stellate reticuloendothelial cells, they both get nutrients and oxygen, get rid of their carbon dioxide and waste materials at the same time. Nutrients are stored or used to make new materials. Poisons are stored or detoxified by the liver, and the blood is returned to the system. It goes to the central vein in the center of the lobule. All these central veins fuse together to form the hepatic veins, which empty into the inferior vena cava. So it says each lobule drains into its central vein. And there it is in the very center, the central vein. And many central veins join to form the hepatic vein, which empties into the inferior vena cava. So let's talk about bile. Bile is a salt. Have you ever talked about bile? They usually call it bile salts. So now you know it's a salt. It's a salt produced by the hepatic cells, used to emulsify fats. EMULSIFY. Fats into smaller droplets to increase the surface area for lipase to act on. What it does is it changes the surface tension of lipids, and they break down from large droplets into millions of little bitty smaller droplets. And then lipase has more surface area to be more efficient breaking down fats. The color of bile is yellow, brown, or olive green due to the pigment bilirubin. Yes, it's a dark green usually. About 800 to 1,000 milliliters per day. So that's like up to a half of a liter, a bottle, a 2-liter bottle. So that's 1,000 mils is 1 liter. pH 7.6 to 8.6, so it's slightly alkaline. And it's going to also be involved in buffering that chyme that comes out of the stomach. So bile will help to buffer it, and the bicarbonate ion helps to buffer that chyme coming out of the stomach. Secretin stimulates hepatic cells and the flow of pancreatic juices. Cholecystokinin stimulates the flow of pancreatic juices and the release of bile from the gallbladder. Now, we'll talk about the gallbladder just a little bit and more in just a second. Functions of the liver. Here are some things that the liver is responsible for. Carbohydrate metabolism, breaking down glycogen to glucose. Lipid metabolism, breaking down fats due to the bile salts. And that's going to be bile salts physically break down lipids, not chemically. Lipase chemically breaks it down. Bile salts change the surface tension so it can't maintain a large droplet. Protein metabolism, so it deaminates proteins, takes the amino group off of proteins. Removes drugs and hormones and detoxifies poisons. Okay. Hepatic cells produce bile for fat emulsification. Stores vitamins A, D, E, and K. Those are fat soluble, A, D, E, and K. And B12 vitamins is water soluble. It stores those, as well as iron and copper. Phagocytosis by those stellate reticuloendothelial cells removes bacteria, old red blood cells, and white blood cells that are old, and helps in the activation of vitamin D. So the liver is very important. There's a little, I forget what you call it, but this is something that has 2 meanings, and it's the life one lives depends upon the liver. So it depends upon the person, and depends upon the liver itself. You can't live without a liver. Looking back a little bit at the gallbladder again, just checking it out right here. Talk about the gallbladder. Pear-shaped sac, which is used to store and concentrate bile salts for a future higher fat meal. Like if you get a steak, it has a lot of fat on it, your bile will be removed from the gallbladder. The gallbladder will contract and squirt more bile into your duodenum. It secretes bile for the duodenum for fat emulsification. Yeah. Gallstones are usually made up of cholesterol, and when they're formed in the gallbladder, when bile enters the gallbladder by way of the cystic duct, the little gallstones will allow it to come in. But when the gallbladder contracts to try to drain the gallbladder to put it into your small intestine for a high fat meal, and so when the gallbladder tries to empty, the gallstones act as a one-way valve. Now it blocks the exit. So when it comes in, the stone is pushed up, and when it goes down, it plugs the hole. And that's why people have to have their gallbladders taken out. Those stones are they're painful when you're eating meals. You have gallstone or gallbladder attacks, and it's kind of painful. Well, let's look on down. The next part of our notes has to do with the intestine, the small intestine. They're called small because their diameter is less than the diameter of the large. And so this portion of your intestine has a smaller diameter than the last part. And so it's called the small intestine, about a 1-inch diameter. The major portion of absorption and digestion occurs here. About 90% of what you're going to absorb occurs here. The other 10% can occur across the stomach, and it can occur in the colon, the large intestine. So the first part of the small intestine, and it's not small intestine, there's no S on there, it's just the small intestine. The first portion is called the duodenum, D-U-O-D-E-N-U-M. Originates or starts at the pyloric valve of the stomach, and it's about 10 to 12 inches long. And that's basically a buffer zone to buffer that chyme with the bicarbonate ion and the bile salts. And then the pancreas secretes new enzymes to help start the digestive process back up. And they'll work in that pH of around 7. The middle portion, where you have about 90% of the absorption that the small intestine is going to absorb, is called the jejunum. J-E-J-U-N-U-M. The jejunum. It's about 8 feet long, and 90% of what you're going to absorb occurs right there in that section. And the last part is called the ileum. I-L-E-U-M. Says joins to the large intestine. So the last part is going to join to the large intestine, the colon. You have absorption of vitamin B12, and this section is about 12 feet long. Now getting back to the lining here, the mucosa of the small intestine contains pits where intestinal glands are located. So yeah, their intestinal glands secrete digestive enzymes. Some examples I gave you are peptide bonds between amino acids, so that's going to break down proteins. Lipase, lipid enzyme, and amylase. Amylose is a starch, and so this enzyme breaks down starches. The duodenal glands, they call them Brunner's glands on some of these pictures, secrete mucus to protect the goblet cells. So you got your little mucus cells here. Some other hormones, enterogastrin. Enterogastrin slows the stomach movements and slows emptying. So enterogastrin, group of enzymes, is going to slow the stomach movements and emptying. Cholecystokinin increases in pancreatic enzymes and stimulates the bile to release the liver to release bile, or the gallbladder to release bile. So increases pancreatic enzymes and the release of bile. You can scratch out enterocrinin. Secretin inhibits gastric gland secretion. So secretin is going to inhibit gastric gland secretion and gastric motility. Villi. This is the small intestine here, and you see that it says villi, small fold-like projections from the mucosal lining of the circular folds. So circular folds goes in your blank. And you see these where it says large circular folds. You can see that the inside lining of your small intestine is folded. And those are called circular folds. And you see that they're covered with millions of little projections called villi. So you have simple columnar epithelium to increase the surface area for absorption of nutrients. Contain capillaries, and we'll look at those in just a second, and lacteals. Now, the first increase in surface area for absorption in the small intestine here is the circular folds. So it's not a smooth lining, It's a folded lining, and that increases surface area. Well, the folds are covered with finger-like projections called villi. And here's a picture of villi here. This is 5 of those you saw covering those circular folds. So these are finger-like projections, and they're covered with simple columnar epithelium. So it says contain blood vessels and lacteals. All right, here's the blood vessels, the arterial and venous capillaries that are going to be supplying oxygen and nutrients to the cells and picking up carbon dioxide and nutrients to take into your liver to be filtered. And then the lacteal is also going to absorb nutrients and fluids from your digestive tract into your lymph system, which is going to be filtered time and time again, scrutinized. So here's the lacteal, this little green structure in the middle, as part of your lymphatic system. Now, these villi, all these villi are covered with simple columnar epithelial cells, and some of those are, let's use the hollow ones, those are goblet cells that secrete mucus. There it is, goblet cells. Those secrete mucus. But what about these simple columnar epithelial cells? Well, let's look at a picture of those, the top of those. Here is one of those cells right here. And they have a modified surface called microvilli. M-I-C-R-O-V-I-L-L-I. Finger-like extensions of the plasma membrane. So it's not smooth on top of those simple columnar epithelial cells. Their plasma membrane has little finger-like folds called microvilli, and that increases the surface area for nutrients absorption tremendously. All right, so we have small finger-like extensions of the plasma membrane of the simple columnar epithelium of the villi, and have tremendously increased the surface area. They're seen under the light microscope as a brush border. So sometimes you can see a little fuzziness on the top of these cells, and that's the microvilli. So the circular folds increase the surface area a lot. They're covered with villi, which increase the surface area greatly, a lot more surface area. And then all the simple columnar cells that cover those villi have microvilli, which tremendously increases the surface area for absorption. So those are all progressive increases in surface area. Circular folds, villi, and microvilli. So your digestive tract is extremely efficient in absorbing nutrients. Intestinal juice, clear yellow fluid, which is slightly alkaline, 7.6. Contains water and mucus. Remember, the mucus by the goblet cells is there again to help protect the lining. This juice provides a medium for the chyme and enzymes to mix. So some of this fluid comes out of your circulatory system by the capillaries. They're going to secrete fluids to help dilute the chyme out a little bit more. Nutrients are absorbed from this solution by the microvilli. And I just gave you some of the enzymes that are in there. It's pretty easy to figure out what they break down. With the first mention of nucleosidases, break down nucleic acids. That's a new one. Segmentation. S-E-G-M-E-N-T-A-T-I-O-N. This is picture B on the right-hand side here. Localized contraction of the small intestine. Circular and longitudinal muscles contract alternately to mix the chyme. You see that you have these two masses, the liquid masses here, one's kind of gray and one's brown. Well, segmentation mixes, it's back and forth mixing to mix it up a little bit more and allow it to come in contact with the mucosal lining to enhance the absorption of nutrients. So nutrients that may be on the inside of these masses, when it mixes it up, now there's more exposure of new nutrients to the microvilli. So it's trying to get as much out of the food going through as possible by segmentation. The picture A on the left, this is peristalsis, the wave-like muscular contraction that forces or moves the ingested materials through your digestive tract. And usually it's one direction. Here it starts at your esophagus when you swallow something. Peristalsis milks that bolus down to your stomach. You have a type of segmentation going on in your stomach, back and forth churning, and then as the chyme leaves, from the duodenum on down is peristalsis. It moves that liquid mass down through your small intestine toward your large intestine. Some digestion. We have carbohydrate digestion, like pancreatic amylase. Amylase breaks down amylose, starch, so it says cleaves starches not broken down by salivary amylase. Now, why would pancreatic amylase break down starches that were not broken down by salivary amylase? Well, it has to do with the pH. When carbohydrate digestion is started in your mouth, the pH is different, right? It goes to your stomach and it goes to pH of 2. You know that proteins, and enzymes are proteins, are sensitive to pH, and so they all get denatured. When you swallow salivary amylase, it's all denatured. Carbohydrate metabolism stops. When the chyme leaves and is buffered from a pH of 2 to a pH of around 7 by bicarbonate ion and bile, then pancreatic amylase can work. It's at a pH that can continue to break down carbohydrates. Dextrinase, you could probably look these up. Dextrose, maltase, maltose, sucrase, sucrose, and lactase, breaking down lactose. Protein digestion. Proteins are broken down to smaller amino acid sequences. Okay, that's what the blank is, amino acid sequences. Now, pepsin, trypsin, chymotrypsin all break proteins down to smaller amino acid sequences. They're all protease enzymes that break down proteins, but they're all different. They don't all break the same bonds of the protein. They are site-specific. So they're going to break proteins down, but they're not going to attack bonds between the same amino acid sequence. They have different sequences. And so peptidases, example, dipeptidase breaks dipeptides into 2 amino acids. And then your digestive tract absorbs those amino acids, and your cells will use those amino acids to build up human proteins. Lipid digestion breaks down fats. Lipase splits fat molecules into glycerol and fatty acids. Remember, bile is a salt. It just changes the surface tension of lipids to where they can't maintain a large droplet, and they break into millions of smaller droplets, which increases the surface area tremendously. For lipase, the enzyme to break down the fat molecules into glycerol and fatty acid, the building blocks that they were built from. Nucleic acids, nucleic acid digestion, breakdown of DNA and RNA by ribonuclease and deoxyribonuclease. Things that you eat, plant material and animal material, contains nuclei. Each of the cells contain nucleuses, so your body is going to derive some benefit by using those nucleic acids also. Absorption of nutrients, essentially all carbohydrates are absorbed as monosaccharides, MONOSACCHARIDES, by active transport or facilitated diffusion. Active transport requires ATP. Facilitated diffusion does not. It just binds and brings it across the membrane. Most proteins are absorbed by active transport, so it takes ATP for most of them. And fats are absorbed by simple diffusion. Now, why would that occur and not active transport? Because fats are lipids, and lipids are soluble in lipids. They'll dissolve through the membranes. Water is reabsorbed by way of osmosis. The cells are pretty much open all the time to oxygen, carbon dioxide, and water. And so water can flow in and out of the cell through channels. The large intestine is the same thing as the colon. Remember, it's lined with simple columnar epithelium. Remember, this from the stomach down to the anus is simple columnar epithelium. It's about 5 feet long. You have the absorption of water, sodium, vitamin B12, vitamin K, thiamine, riboflavin, and part of that's due to the E. coli, the bacteria which is in there, which helps to continue digestion. So E. coli was named, the coli portion has to do with colon. So it contains the bacteria E. coli, which further digest the chyme to produce vitamin K and some Bs. So there is a benefit to having the bacteria in your large intestine. Now, where the small intestine joins the large intestine, there's a little sac there called the cecum. C-E-C-U-M. It means blind sac or pouch at the junction of the large and small intestine. And there's a valve between the small intestine and the large intestine named after that. It's called the ileocecal valve, named after the last part of this small intestine, this joining to the large, and that cecum. So it's called the ileocecal valve, a sphincter between the ileum and the large intestine. So the ileum will squirt its chyme into the large intestine after it passes through that ileocecal valve. It opens up and lets the chyme go into the large intestine for the last processing to occur. You see that vermiform appendix. We usually just call it the appendix. Just like when you were talking about the pons, you didn't say the pons varioli, and the medulla, you didn't say the medulla oblongata. The same thing for this. Usually it's just called the appendix. A small tube about 3 inches long, which hangs from the cecum. Now let's continue with some things that are not in your notes here, but it's, you might want to add them. You have the ascending colon. On this left side, you see the word ascending colon. It's kind of blurry, but it's after the cecum, it goes up on that right side. It's called the ascending colon. Where it bends to go across the front of your abdomen is the right colic flexure or the hepatic flexure. So it's bending. Then it goes across your abdomen as the transverse colon. It's not labeled there totally, but it's called the transverse colon. It bends again, the left colic flexure, or the splenic flexure, because the splene's on that side, and it goes down, the descending colon. Well, it has to go back medial, so it bends, goes medial, and bends again. That forms a lazy S, and the word for that is sigmoid. It means S-shaped. So this is the sigmoid colon as it bends, goes medial, and bends again to go to the rectum. That's the sigmoid colon. The rectum is a muscular storage area for fecal material that has been dehydrated and is ready to be eliminated from the body. It's going to be stored in the rectum. And then the last portion would be the anal canal. So let's look at, I have some things here labeled here. There's your ascending colon, your right colic flexure. Here's your transverse colon. I got it boxed right there. It's transverse colon, left colic flexure, descending colon, sigmoid colon, then the rectum. Some more structures. These little pouches, you see the little pouches all along the large intestine. Those are called Haustra. So let me go back to 16 for just a second. Yeah, back here. Yeah, we'll look at this one. There's the Haustra. So going down your notes a little bit, it says Haustra, pouches formed along the length of the colon. Haustra. And then right above that is Teniae coli. There's a band of muscle goes down the middle on the front and the back of the colon. It's on the right hand side there. Teniae, second word coli, longitudinal bands of muscle. Let me go up on your notes just a little bit. Here's the anal canal, the last inch of the gastrointestinal tract. The last inch, anal canal. So the rectum's up here at the top. This is the anal canal, and it's surrounded by two sphincters, the anal sphincters. There's an internal anal sphincter, and that's smooth muscle. You can see it there. And the external anal sphincter is a lot larger. It's skeletal muscle on the outside. So there are two sphincters, internal and external anal sphincter. Now hemorrhoids are varicose veins of the rectal veins. So here are the hemorrhoidal veins. These are the ones that will form hemorrhoids if you strain real hard when you're going to the bathroom, like constipation or even childbirth, can cause these to billow out and cause hemorrhoids, which are a little painful and itchy from what I understand. Two more definitions, well, several more. Defecation, the elimination of fecal material from the body, is called defecation. Diarrhea, defecation of liquid feces. Dia means, well, the diarrhea means to flow through, is what the word means, to flow through, and it is liquid material. Constipation, infrequent or difficult defecation. So those are some definitions that are already in your notes. Here's another one you need to add. Crohn's, C-R-O-H-N-S, is an inflammatory bowel disease. And you can look that up if you want to check it out. See what that is. So it's not irritable bowel syndrome. So you could look up the definition and see the definition between the two. This slide is showing some of the mesenteries, the serous membranes of the abdomen. And if you remember, I said the mesentery supports the small intestine. So you can see on the left picture, pointing out mesentery, but look on the right. And you see that we have mesentery and it's pointing. That's a double-layered serous membrane. And you see it's attaching this small intestine to the back of the abdominal cavity. And these little, looks like little tree parts coming down here, that's the mesentery encircling portions of the small intestine that have been cut through there. So the mesentery supports the small intestine. The transverse colon is supported by the mesocolon, and you can see that on this picture on the right also. There's your large intestine, and you see that it's got the mesocolon holding it. Now, something else, when you stand, babies are born, their spine is pretty straight. And when you stand, you start standing, your organs start to fall forward. Your small intestine and your large intestine try to fall forward, and that's what puts this lumbar curve in your back. Because see, the mesentery is attached to the lumbar area of your abdomen, back of your abdomen, and so is the large intestine. So they're going to pull on these vertebrae and put that curve in there. I have some more slides, functions of the mouth, esophagus, and stomach. You can look at that on this PowerPoint, and you can look at the continuation, which is right down here, of the functions of the large and small intestine. Okay, so I gave you two slides to look at that. This one and this one. And they should be on your PowerPoints also.